Okay. All right, we're going to talk about denaturing proteins. Denaturing is when you take the protein apart or break the protein. So, what kind of things cause proteins to break down? Okay. Um, Sometimes they don't break all the way down, or sometimes they'll break down radically all the way. They're not coming back. Sometimes they can come back. A really good example that you all know about with denaturing proteins is egg whites are proteins, and they contain proteins. When you drop an egg onto a hot frying pan, that protein denatures. It changes from a liquid to a solid, it changes its color, and the yolk does too. So it's not just the white, but the white seems to change the most. And that's an example. It's not like when the egg gets cold again that it turns back into a liquid, right? So, what are some things that break down proteins? Big shifts in pH. Okay, that's just going to chemically pull the protein apart. High changes in salinity. So, all those ionic forces are going to change the shape of the protein. Heat. Now, when we say heat, I want you to remember that different proteins and different organisms live at different temperatures. So what temperature would you think most human proteins are like Goldilocks? They're going to work best at what temperature? Yeah, 98.6 Fahrenheit, 37 degrees Celsius. So you start getting a human being out of that Goldilocks temperature, and their proteins don't work as well. That's why high fevers are, well, one reason why high fevers are so dangerous. So proteins work in the environment they evolved in, okay? So are there proteins that work in extreme pHs? Yes. Extreme temperatures? Yes, okay? So don't think that everything needs to be like humans. There are bacteria that can work in really high salt environments. They're called haliophiles, I think. So here's what happens. We have this really well organized, that's the intended shape. You get it out of its Goldilocks chemical zone, and it starts breaking back down. Sometimes it doesn't break down so far that it can't go back together. So they call that it denatures or it renatures. Okay, so if it's not broken down all the way, we can put it back together. This is kind of like you dropped your Legos and you just need to put it back together a little bit versus absolute destruction. You melt your Legos. It's not coming back. Okay? Now, this gives us opportunities to do some pretty cool things, okay? Because there are so many amino acids, and there are some that are out there that aren't found in eukaryotes, that are in plants and so on, bacteria, we can change different R groups, okay? We can give proteins entirely new properties. And so, genetic engineering, we can either use bacteria to do that. So you just inject a ring of DNA into a bacteria that's like, make this amino acid. Make this protein out of amino acids. And we do that in this class. We'll take E. coli, a safe version of E. coli. We'll heat it up and then cool it down to make its cell walls crack open. Then we'll shove a bunch of DNA rings into it, hopefully. And when those DNA rings get into it, the bacteria start saying, first of all, they have to be turned on by a certain sugar. You have to say, only make this under these conditions, and that's how a lot of protein, it doesn't just make it all the time. So you switch on that chain of DNA and you say, make this protein. And the protein we tell the bacteria to make is a glow-in-the-dark protein in jellyfish. Now, we choose that one because it's so easy to say, we were successful, we did it. All you got to do is shine a black light on it and it glows. If we said, we're going to put a ring of DNA in there that says, make, I don't know, the protein found in your eye lens. Well, that would be really hard to confirm. Like, well, how do we say we did it? So that's why we use the glow in the dark. Plus, it's cool, okay? So this has been done with rice, okay? There are areas in the world where rice is sort of the main carb that people eat. And in many of these places, they don't have enough beta carotene in their diet. So beta carotene, nutritionally, if you don't get enough of it, there's health problems. One of the main ones is blindness, okay? So you can kind of hear the carrot. What does everybody say about your eyes and carrots? You got to eat them. You'll be able to see good. So an organization, I can't remember if it's called Golden Rice or Yellow Rice. They have set up this genetically modified rice by inserting instructions into the cells to have this particular substance made by proteins. This could solve a global health problem. Some people are a little conservative about genetically modified organisms, okay? When we say genetically modified, it's not like we're telling this to make something that's never been found in biology before. But some people will say, well, what happens if this enters the sort of, what happens if this enters not agriculture, but the wildlife ecosystem? There could be really bad consequences. So there are two sides to the argument. I tend to come down on things that make less people blind are better. I mean, I think that the risk, personally, I think the risk is worth it. There are some sort of like science fiction-y kind of future-y ideas out there about, let's make bacteria that can eat oil spills. That sounds good, right? What if that bacteria gets into the wild and like starts, I don't know, infecting gas stations and they eat all of our gas? Oh, there's too much plastic in the ocean. Let's make bacteria that can break down plastic. Sounds good, right? So what if that gets into the industrial system and the consumer side of things? Your shoes are made of plastic, your phone's made of plastic, and you look down, you get mold on your bread or whatever, or you get like a bad gallon of milk. What if your like plastic things in your home like sort of rusted away from these bacteria? That would be bad. I mean, it would be good for the oceans, but we use plastic for a lot of stuff. Artificial heart valves. Imagine you got that bacteria in your body. All right. So I'm gonna use this as my stopping point for today, and I hear the bell, so we just timed that perfectly.